Okay. So yeah, thank you for the invitation. The work I'm going to talk about today is, as in the title, going to be on understanding the inheritance of non-genetic um, heterogeneity in gene expression states. And all of the work that I'll talk about is in collaboration with Shaun, who you heard from yesterday. Uh, but before I talk about that, I guess Yogesh insisted that I put a slide in about Waddington landscapes. I have to, I have to pay my respects to the organizers, so I.
3 slides.
uh, oh, 3 slides.
One for each
I only
organizer.
did one. I'm sorry, but I thought at least I'd put in some other pictures of the landscape just for variety. In fact, the one on the top right there is from Joseph Needham's book. That's 1936. On the left is a collaboration he did with an artist in 1940. And then this one is the middle one is also from the Strategy of Genes in 1957. But obviously, this was a long period in which he developed this idea of a landscape. But um, uh, just to make a couple of points that I think, in many ways, one of the difficulties in interpreting Waddington's landscape today is not simply a technical problem, but also, I think, a philosophical difference that Waddington had with the way, as he saw it, a kind of more mechanistic conception of biology. But I think there are ways to argue that the Waddington landscape is an early example of what we would call an emergent description. And in fact, the language of emergence, when it entered into theoretical physics and condensed matter physics, took the language is actually taken from biology. And if you actually look at the history, you actually even read Anderson's works. He himself says that actually this is a language that was earlier used in biology that then was adopted into physics and, of course, very successfully uh, uh, turned into a much more theoretical idea. But just this, right, this exchange between Crick and Waddington is always good to read, where Crick was not a fan of the landscape, and they have an exchange of letters. And one of them, he said, "Of course, one grasps in a rather vague way what you mean to convey, but I found it difficult to make the analogy precise." And Waddington simply was like, "It's a simple and perfectly clear idea." So if you don't understand the landscape, that's his answer. But I won't talk about the landscape. I think Eric is going to talk about it later. I'll talk about something else, which I thought would also be of interest to the audience here. And the motivation for it is the fact that cell fate is is correlated across lineages, and so one of the things that Shaun saw in his previous work is that if you take a bunch of cancer cells and you expose them to drugs, then sister cells tend to share fate, which means that there are lineage correlations which are important for the kinds of fates that cells take. And of course, Yogesh has been involved in work where they have this right idea of memory genes. I'm not sure how you came up with the term. I'm not sure it's the best thing to call it, but anyway, we'll stick with the term, the idea of memory genes, but genes whose gene expression states are heritable across a few cell divisions. And what you can, and presumably, and possibly these memory genes are important for cell fate, that certain gene expression states may make you more, may make you able, might make it possible for you to survive, for example, drug treatments. And one way to discover these memory genes is to do a kind of Luria-Delbruck-like test where you take several replicates, you look at gene expression and you look at its variation across replicates, and that, right, if you have a gene which is fluctuating quickly, then you don't right. Then the distribution that you see across replicates is sort of narrow, whereas if it's fluctuating slowly, then you expect a wider distribution in gene expression across replicates. So if you had lineage information, then it is possible to say something about the dynamics of gene expression, in particular, whether right, gene expression states are being preserved across a few cell divisions. Okay, so the question that we sought to ask was kind of a practical question, which is, can you do this without knowing lineage information from a single snapshot, single cell RNA-seq data set? And even though it was a practical question, it turned out to be an interesting theoretical question and sort of made us think about the problem a little bit differently, which is what I want to tell you about today. Okay, so this is sort of part of this broad question of how do you get dynamic information from single snapshots. But unlike some of the other things that we've already heard about, pseudotime, RNA velocity, and so on, here the setting that we are interested in is to think about gene expression states which are, let's say, around some steady state. But as cell divisions are happening, these genes, so actually let me introduce the site. So you imagine some gene expression that is around some steady state as cell divisions are happening, which are these D0, D1, D2. Suppose there's a fluctuation in a gene expression state. The question is, what are the time scales on which it relaxes back? And if the time scale in which it relaxes back is less than a cell cycle time, then this gene is not a memory gene, right? It relaxes very fast. If the time scales on which gene expression relaxes back is longer than cell cycle times, then you expect that, for example, sister cells and cousin cells will have certain correlations. Now, of course, here I've drawn it as gene expression, but really what you care about is the autocorrelation. So really, you can think of this as the autocorrelation. Just as a cartoon, I've actually shown the gene expression going down as. Here also, I'm assuming the gene expression is perfectly inherited at cell division. I can come back to that at the end. That actually ends up being important. If you start introducing division noise, that leads to some important consequences. But in this setting now, so you have a certain set of genes, right? You can basically talk about their half-life, where their half-life, if you measure in the scale of cell cycle time, then tells you what are the number of cell cycles for which your gene expression remains correlated, and then gives you basically a measure half-lives that are greater than a few generations are examples, right? These genes are sort of examples of memory genes. Okay, so yeah.
How do you normalize the y-axis? Because it's going to change everything, right? If you're measuring fold change or absolute numbers, or
right.
I just take your eye.
Right. So I mean, okay. So this is normalization, which let me just get back to the normalization question. How do you normalize the actual data? So here, the idea.
Are you thinking about fold change, or are you talking about absolute?
So we're thinking about concentration, not about numbers. We're thinking about concentration. But in practice, what we're doing with the data is that we're sort of dividing. So we're scaling by, right? So in the single cell RNA context, we're scaling by the total UMIs. So we're sort of dividing, right? So we're scaling the data. We're also actually, in the end, normalizing it by dividing by the variance and subtracting off the mean. So it's actually, in the end, it ends up just being a z-score. But yeah, we can also get back to how we're normalizing the actual single cell RNA data set. Yeah, that's an important, yeah. That actually is sort of
conceptually, you think biologically it's the concentration that's biologically important?
That's biologically important for what? Depends what you mean.
Memory.
Well, for memory, meaning so two things clarify. I mean, if you just scale the data, right, we are talking about the time scale at which it decays. So the scale doesn't matter in that sense.
So you transform it.
Yeah, but but what do you mean? Okay. Okay. No, so anyway, so the question here, so actually you could actually talk about absolute numbers. So again, I'll maybe get back to that at the end. You could just talk about when numbers are inherited, but here we think about the concentration and the time scale actually of the time at which the gene expression decays actually doesn't matter. An absolute scale there doesn't matter. Okay, but the main idea about how we can get dynamic information of this sort, which is the dynamic information here is, right, what are the timescales in which gene expression fluctuations decay from a single snapshot is sort of represented in this cartoon. So suppose you have a cell, right, that divides, and you have here, I'm just showing 4 cells that have some, so these are some lineage correlations. If my gene expression states decay very quickly, then I don't expect any correlations in the cell. And so if I look at the cell-cell covariance matrix or the correlation matrix, in the end we just look at, as I said, normalized things. The cell-cell covariance matrix has an eigenspectrum that actually kind of looks flat. And I'm going to draw these kinds of graphs a lot, where I look at log lambda the eigenvalues of the cell-cell covariance matrix, and I just sort the eigenvalues and I plot log lambda versus rank. On the other hand, if you actually have right, these kinds of memory genes which are slowly decaying and so lead to correlations between sister cells and cousin cells and so on, then the cell-cell covariance matrix has a more specific structure. right? Where sister cells will have some correlations, cousin cells will have correlations that are somewhat smaller. And what that leads to is a wider distribution of eigenvalues. The important point here is that actually in this gene cross cell matrix, I can rearrange the cells and the eigenspectrum remains unchanged. The eigenspectrum of the cell-cell covariance matrix remains unchanged. And so you actually don't need lineage information if you're just looking at the eigenspectrum. And the point here is that this kind of wide distribution of eigenvalues is actually a signature of having lineage correlations in your data set. Yeah.
Can I just push back on that a second? Yeah. That it's a signature of many other things as well.
Sure, it could be. Okay. I agree. Yes. It could be a signature of many other things. And in fact, I'll also add to that, in fact, the cell-cell, because it's a single sample that we're going to be working with, the cell-cell matrix has exactly the same eigenvalues as gene-gene matrix because they're just a transpose of each other. So in fact, the two effects get mixed up. So what we're going to try and find now, I'm going to show you a more specific signature of what we think lineage correlations will do. And of course, there could be things from gene-gene or other ways, spatial signaling, for example, that could look like that. Yeah.
Just to sort of confirm, you're saying if you have these big band gaps, that is characteristic of lineage correlations, or are you saying something that looks like power law?
A power law, yeah. So I'll talk about the power
not the band,
in a second.
Not the fact that you just have a band gap.
Yeah. So this is just a cartoon. I'll show you the calculation.
Yeah. Sorry for missing this, but it seems very important. What's the difference between C and D?
C and D. Sorry, these are just divisions. These are cell division.
Figures C and D.
Oh, sorry, yes. Figures C and D. So the idea is in C, you only have these kinds of non-memory genes, which all decay very quickly. And so there are no correlations between cells, even though the cells are related by lineage. Whereas here you have some genes that do have gene expression states that are correlated in sister cells and cousin cells, which are reflected in the structure of this matrix. Okay, so this is kind of right to give the idea, but we'll talk more specifically about what the problem we have to deal with. So what I showed you is the idea of if you had an ensemble and you looked at the true covariance, and you actually knew the lineage relationships, and that's what the cell-cell covariance matrix would look like. In practice, you have a sample covariance matrix. You don't know the lineage relationships, and so what you get is something that just looks like noise. Question is, can you still extract some meaningful information about the time scale of gene expression decay from this kind of sample covariance matrix? And so the way we calculate that is by using a random matrix theory. And the basic idea is that the null model for data is noise, which we've heard a lot about how important noise is. And so you can just ask, if I take a gene cross cell matrix and I just fill it with noise, I sample that noise. It actually doesn't matter what distribution you use, more or less. And the distribution of eigenvalues in that case is known to have a universal distribution. It's called the Marchenko-Pasteur distribution. This is a well-known result. And so in a certain sense, the Marchenko-Pasteur distribution is sort of your null model. If you take a large random matrix, you just fill it with random elements. What does the spectrum look like? And of course, there are all sorts of correlations within the matrix because of gene-gene interactions, cell-cell interactions, and so on. The idea is that deviations from these distributions are informative about what those correlations can be. And so what we're doing is, in a specific model of lineage correlations, we're going to try and calculate how this distribution will change because of the presence of lineage correlations in the data. And so we can do that calculation. I won't go through the calculation. For those who are interested, I've just put in sort of, a summary. This is a way to calculate the distribution of eigenvalues for correlated matrices was already known to Marchenko and Pasteur. What you have to do is you take the true covariance matrix, you calculate its eigenvalues, and you can find out what the distribution should look like for the sample covariance matrix, which is a summary is that the sample covariance matrix, again, you have a single sample, you look at its eigenvalues. The sample covariance matrix is a systematically noisy version of this true covariance matrix that I showed you as something that you can calculate. So what we can do is we can make a model where we just introduce lineage relationships and we can ask what kind of distribution of eigenvalues will you see for large matrices where the only relationship is because they're lineage correlations, because some genes are correlated in sisters and cousins and so on. And so we can make a numerical simulation and we can ask what that looks like. And basically the eigenvalues have a power law form. So again, if you just introduce lineage relationships and everything else is just, and you look at a noisy sample, you get an eigenvalue of the cell covariance matrix, which has a power law form. The power law is in the eigenvalue as a function of the rank. Again, so this is just showing that we can do numerical simulations and we can actually compare them with analytical results. If your cell cycle time, so in our numerical simulation, if we make our cell cycle time very large and keep everything else fixed, which means you no longer have memory or cell cycle because everything is measured in terms of cell cycle time, then you just go back to the Marchenko-Pastur distribution. So these power laws, we think, are a signature of cell-cell correlations that come as a result of lineage correlations. Okay, yeah.
Quick question: did you consider the dispersion over time of the transcriptome? Because we measured that, and it's like after 5 cell division,
yes,
the daughter cells they drift away, right? Is that, how do you consider the expectation of similarity of daughter to the mother?
Yeah, so that's not something we consider. So we don't consider that kind of drift. It is something that we are thinking about now. Even in other contexts, if you had a changing transcriptome along with this, then how would that change us? In this, we've not considered that. Yeah. Yeah.
So people have written down models for evolution of continuous traits on phylogenetic trees, and the covariance matrix for the measurements on the leaves will reflect the phylogenetic distances of these cells. So that, have you looked at how your work relates to that part of literature?
I mean, maybe we can talk offline about exactly which literature you're referring to.
Okay.
Yeah. Okay, so that then actually allows us to make we thought of many ways to try and actually identify so how do you now identify genes? So the idea here is that lineage correlations lead to the specific signature of a power law on the eigenspectrum, and then we came up with a kind of very simple method to try and identify these memory genes, which is simply to take your original data matrix. So again, here now I'm going to show you the results for single cell RNA-seq data. And again, in terms of normalization, what we write, we scale for sequencing depth, and then we actually just take z-scores. So we actually scale everything by the variance, and we, as I said before, we subtract off the means. And so in the end, you're just looking at a cell-cell correlation matrix, and you're looking at a eigenspectrum. And now you just remove a gene from the data set.
You scale the mean and variance of the gene across cells?
That's right. Yeah. Mean variance gene across cells. Okay, and so we just remove a gene and then we just look at how it affects the eigenspectrum. That's another thing we can actually, in the context of this simple model, we can analytically calculate and find that actually you can get statistically significant results when you remove a slow gene, meaning when you remove a memory gene. And when you remove a non-memory gene, then basically the spectrum remains the same. But when you remove something that's actually a memory gene, then you can see significant deviations in the spectrum. And we can use those to try and rank genes by how much they're basically leading to this power law. So that allows us to kind of make an algorithm to identify these memory genes. And so I'll just show you the results. The first is we took the same melanoma cell line in which previously the memory genes had been identified because we had ground truth there. First of all, you do see these kinds of power laws. And you can just shuffle gene expression across cells to write as a control to see that destroys the power laws. But then more importantly, you can actually just from this single data set identify these memory genes that had previously been identified, right, with several, I forget the exact number, but 40 or 50 replicates. What you can ask for is the cumulative distribution function of identifying genes, assuming that this original data had ground truth, and that rises very sharply, but you would expect it to go linearly by random. Yeah.
I'm going to be a little bit annoying. Can you go back to the last slide?
Yeah.
I mean, don't we learn that the red genes are probably not memory genes, and the green genes are memory genes or something else?
Yeah. I mean, so again, the question is, whether these power laws could be produced by something else.
Yeah.
We're looking at cell lines in a dish. I mean, maybe they could be produced by something else, but I would say that it's a little difficult to get power. It's not so easy to get power laws. You need to tune some things typically to get power laws, whereas in this kind of lineage framework, it's just because you have the division process, the power laws come about kind of naturally. Yeah. I'm not saying that there couldn't be. I'm just saying that it's a little difficult. Yeah.
If you have state diversity at baseline,
yes,
like different states,
that's right. Yeah.
like itself, plus also like your culturing, and there are some local effects that give you a subset of populations with particularly defined phenotypes. I mean, I'm having a hard time justifying using this method in real data.
Yeah.
As opposed to, let us say, just look at the marker genes of the population that are going to be constant. Like, I don't know what do you gain, and I think that the assumptions for it to work rarely are going to be present. Like, there's always going to be environmental drivers and maybe even like some gene regulatory network action that's like switching the states. I'm not sure. Like, do you think like coupling it with barcoding and looking at that?
So, I mean, what's interesting, I won't have time to talk about it, but what we've discovered now is we can actually find lineage relationships using this and compare it to barcodes. So we can just take single-cell RNA-seq data and we can identify lineage relationships that you would otherwise need barcodes for. And obviously, it's not perfect, but that's also giving some evidence that what we're discovering is not just something else, but has something to do with those lineage relationships. So those kinds of tests we are doing to see that. And then in terms of cell state heterogeneity, I mean, what we've done, for example, if you have tissue data, is you cluster and you remove, right? You take a subset of cells. That's not perfect. Ideally, we would want a theory where you can have cell state heterogeneity. That's something we're working on. If we don't
also, like my follow-up question is something you said at the beginning.
It's not cells, but the different cell types, cell type heterogeneity.
Or even like cell state within a cell type. When you discuss the idea of like as random assortment of the contents of the cytoplasm upon division, no? Like there's a whole literature in the origin of life about protocells and like how that could impact. Like, have you considered that for your?
Yes, I won't have time to get to it, but we can talk about it. Yeah, about how the assortment. Actually, it's quite interesting how that affects some of this. Yeah.
Sorry, just a very quick question. So, if you sort of invert this back into the variance is explained by the different principal components because you're doing CC transpose. What do you see that there are a few drivers of all the variants?
So that's interesting. I mean, so again, the cell and gene. As I said, because a single sample, they have exactly the same eigenvalues. The eigenvectors in one case are in cell space, in the other case in gene space. The gene space eigenvectors actually don't seem to have information. They're corrupted by the cell-cell correlation. So if you look at these eigenvalues and try and interpret the gene space eigenvectors, I mean, we can check in our model that they don't actually have information about the genes. So that's the first thing we try to see. And that's basically because of this problem that you have the same matrix. And so it's kind of difficult to interpret exactly one or the other. They kind of get mixed up. Yeah.
So if you plot the variance of the green gene versus the variance of the red genes, like how do you rank in the rank variance spectrum? Like if I were to like filter the data matrix from some sort of feature importance through variance, am I always getting rid of the fast non-memory genes and retaining the memory genes, or?
No, I mean, so we tried stuff like that. We tried, for example, looking at the coefficient of variation and just ranking by that. None of those seem to be predictive with memory genes in the same way.
How is it different from David Souter's work? But can you just quickly tell us?
Well, so in David Souter's work, which we've looked at quite closely first, it's actually ultimately their algorithm's kind of heuristic. The data set that they originally used there, they have replicates again. They originally used to identify memory genes, but then they come up with a more kind of heuristic algorithm. We think it works better than theirs, but we can talk about it. Yeah. Okay. Yeah.
What's the computational time of your algorithm? Because it seems that you need to fit around the plasticity.
Yeah, I mean, it takes about 2 hours. Yeah, you take a data set and but you have to do it once and then you get all the, but it is a bit computationally expensive, you're right, yes. Okay, I guess I'm out of time, right? Okay, so I will then skip over, I guess I was just going to say that, you actually a lot of these ECM related genes seem to come up. We've seen this now in many, many data sets. And then I was going to go over this, but I'll skip this. But there's some nice ways you can theoretically think about cell division partitioning and what kinds of gene regulatory networks will lead to this kind of memory. But I'm going to skip that and end in case there are any more questions. Yeah.
Feature plus.
Very nice, thank you. Is it fair to say, from a dynamical systems perspective, that a memory gene is one slow relaxing direction?
Yes. So in fact, what I skipped over is basically, right, actually there's a network. So actually there are really eigenvectors. And there's some modes rather than genes. I mean, there are actually some slowly relaxing modes. And what we have is then the question becomes, what's the eigenvector of the slowly relaxing modes and which genes are represented in it? And we think we have a way to try and calculate that, yeah.
And when you say slowly relaxing, that's just with respect to the cell cycle time, right?
That's right. Yeah, that's the time scale. Yeah.
Thank you.
All right, thanks.